Harvard wanted it for various analytical purposes. I wanted to get a decent amino acid analysis. We had learned that what had been published as pure phytochrome with a molecular weight of 60,000 was in fact just a proteolytic fragment, and that it was much, much bigger. And we had bigger stuff, and so I wanted the amino acid analysis. Elaine was doing biophysical stuff. Both John and Harvard wanted antibodies, and so on. All these things, different things. And we really did have very serious discussions, because any one of us could have used all of it. And we were doing it around every 2 weeks. Well, things didn't always work well. There was a fraction collector that was the last step. Oh, by the way, I got up at 4 in the morning to get down to run the DEA column, because things were going continually from Monday morning through sometime mid-morning Wednesday. And so the last step was a P150 or a G200 column, very slow. So that was set up and run overnight. Well, one of the graduate students, who was pretty fatigued at this time, was responsible for that. He set it up and turned the fraction collector on, but he'd forgotten something, namely tubes in the fraction collector. So we came in the next morning and there was this pool of bluish solution on the floor. Well, another graduate student said, "Oh, let me do that next time." Actually, he didn't use that tone of voice. So that was fine. So 2 weeks later, it was agreed that he would do it. He did. He put tubes in the fraction collector, but guess what? He didn't turn it on. Things were very quiet. Another time, the brushite, we had a 2-syringe pump that we were pumping calcium fluoride and calcium phosphate to make the brushite. Well, sometime early in the night, and this all went into a big garbage can, sometime early in the night, one of the tubes popped off. So the next morning we had the most remarkable slurry on the lab floor. And then, well, I wasn't having good luck with the DEA column, so I went off to Jerusalem, and while I was there, somebody else would have to do it. I got a telegram just before I gave my first talk saying the DEA column works, stay in Israel. Okay, a couple of wrong turns. Bill Curtis came to do a sabbatical and started studying phytochrome phototransformation. And by gosh, look at this. If you do a log plot against the light dose, you get a curve that looks like that. And if you now subtract this straight line from that, you get a second. So this is 2 components, very rapidly converting and slowly converting, and in both directions. But this was exciting because we knew that there were some very sensitive phytochrome reactions and some other ones that weren't so sensitive. By the Lord, we had the answer. So anyhow, we published a paper, and then I was in a meeting and I met a guy by the name of Bob Decker. And Bob Decker was the person who had built the special, the very special spectrophotometer that we used to monitor phytochrome. So he said, "How are you doing?" I said, "Well, no, we have this really exciting finding. You know, we have the 2 components, one that's more sensitive than the other, and it's going to be fascinating to find out what actually accounts for that, and where they are in the cell, and a few things like that." There was a long silence, and he said, "Well, I should alert you that that instrument has a switchover point where it changes its sensitivity." Oops. So we repeated the experiment and used a different spectrophotometer, and guess what? We had published on an instrument kinetic, not a phytochrome kinetic. So we published a retraction. This was both true in vivo and in vitro. So we published a retraction, but hey, we got 2 papers out. Well, anyhow, back to Stanford and Carnegie, and that was interesting too. I won't go into the details, but Phil Abelson had interviewed me, who was the president of Carnegie at the time, and asked me what I would do with the Department of Plant Biology, because Stacy French was retiring, and they were superbly equipped for physiological ecology, superbly equipped for doing physiological ecology, but there was nothing really to do good biochemistry or molecular biology. So it really needed to get pushed in that direction. So I told him that I would recommend that they arranged things so that that could be done. And the physical plant was not in very good shape, so I said, well, what you need to do is invest $1 million in capital improvement, double the annual budget. And Phil said, yeah, well, that's what we have in mind. You want to do it? Well, there was no question. So then we discovered something else. We didn't discover it. Somebody else discovered that if you turn light onto blue light onto a plasma membrane preparation, my gosh, the cytochrome went reduced. A B-type cytochrome. Here's a different spectrum. So when light on is dark here, and you notice there's a big shift in the striated region of the spectrum, and the alpha band comes up very nicely. So we studied this, and we got paper after paper after paper. Yeah. However, we never could prove or demonstrate that it had any relevance to biology. And then when Warren Butler and his colleagues showed that you could take horse cytochrome C and riboflavin off the shelf, mix them together and turn the light on and reduce the cytochrome, we finally reluctantly concluded that this LIAC, or light-induced absorbance change, remained a completely enigmatic reaction that didn't have any physiological role. Well, but again, hey, 7 papers. Well, other topics again, I won't go through them all, but we've gone back now to that mode, having spent so much time together on phytochrome at Harvard. Lots of other stuff going on. And back to blue light, and again, just like Jen Wilson, Sean Gallagher changed the course of events very dramatically, because he discovered the phenomenon that we now know as the light-activated phosphorylation of a plasma membrane protein. And Tim Short was in Peter Rhee's lab, and fortunately for me, Peter wasn't interested in going on with this project, and generously turned it over to me. Otherwise, Peter would have worked on it, and that's fine. He would have done a terrific job. But it was handed to me on a silver platter. And so, progress, we got a mutant that was impaired in Arabidopsis, that also was impaired in phosphorylation. That was a nice connection. And Matty Liscombe then produced a whole series of phototropism mutants. We finally identified the gene for the phosphoprotein, and it was a serine-threonine kinase with these 2 domains upstream that were almost identical, which we named love domains because they looked like domains in proteins that were light-oxygenated voltage. And then finally, John Christie expressed the protein in insect cells and showed that it would do the photochemistry, and that the flavin was, the chromophore was the flavin. And then he showed that the binding sites were, the love domains were the binding sites, and then we demonstrated that the love domains were photochemistry. Well, that's sort of a quick summary. Here's the structure of the thing, and here is from Kevin Gardner's laboratory, Shannon Harper's very nice NMR solution structure. Here's the flavin, here's the cage for the love domain, and here's an alpha helix downstream. When we turn the light on, the blue light on, there's a formation of the cysteamyl adduct between the protein and the flavin. That tug releases this, and this guy flips out, and that apparently activates the kinase. And this is photochemistry. I won't worry about that. But it took us 41 years to identify one measly blue light receptor. Now that isn't exactly roaring progress. So if your stuff isn't going very well, don't worry. There's plenty of time. Look, so some people are slow.